So are you guys understanding what I'm saying, what I'm teaching to you?
Yes, ma'am.
Thank you. Okay, so now the SAM format. In the SAM format, you can see it's the full form of the SAM format is sequence alignment map. What it is? This format describes how your short sequencing reads map or align to long reference genome like FASTA. So here you can see for what reason actually the SAM format is used. When you have the FASTQ files, right? You have the read sequences, and when you want to align those reads to the reference genome, now you want a format, a file format where you can understand are these reads mapping to the reference genome or not. To view everything in a file, so there is a format called SAM, sequence alignment map, which will show you how many reads are aligning to the reference genome and how many reads are not aligning to the reference genome, right? This SAM format is a readable text file. You can read it, you can understand the SAM format very well. File extension which you have to use is file.sam. Here the extension is .sam, right? Then here you can see this is the file of the SAM. I'll zoom it, right? So in the file format, you can see here there is a headline. So here you can see there is a headline. This is which starts with @ SQ and PG, both are the headers. And in this information, you will get the information related to the reference genome, which reference genome you have used to map, and the length of the this is the length of the reference genome. These are the PG is the program used in this mapping. So BWA program has been used. The version is this. Command line has been used, BWA mem reference.fasta, and this is the R1. This is the FASTQ file. So this FASTQ file has been mapped on this reference.fasta. Here, since you have read Illumina and other sequencing procedures, there are paired and reads also. So here I have just mapped only one read sequences on the reference FASTA. If I use two read sequences, that can also be possible to map on the reference FASTA in the same file. Now, in the first column, you have to observe it properly. So in the first column, what you will get? You will get the read ID, the FASTQ ID, which comes after @. So you will see here the read ID. Then in the next section, you will see the flag. This is important. Flag. This flag is representing if there is zero. If there is zero, it means it is it is forward mapping, right? Sometimes I also get confused. So zero means it is forwardly mapping on the reference genome, and if it is 16, it means that read is mapping on the reverse side of the reference genome. So you will get 16. And if you get a number 4 here, it means the reads are not aligning. They are unmapped. Now this is in the third column, you can see this is the ID. This is reference ID on which this particular read is. Yeah, yes.
Ma'am, 0, 16 and last. What was it?
0, 16 and 4. 4.
4 is what?
Okay, I just zoom out this. So here you can see 0 for forward mapping, 16 for reverse mapping, and 4 for unmapped.
Okay.
Got it? So
Yes.
I have zoomed this just so that you can see the file properly. That's why I have zoomed it. So here you can wait. So here this is the ID, this is the flag, then this is the reference ID on which this particular ID has been mapped, and then you can see this is the position where this particular ID has mapped on reference genome. So this is the position on the reference genome where this particular read ID has mapped. 60 is the mapping quality, which is the highest quality, and 100 m is the matching. How much? This is the cigar unit. This is the cigar unit. It means how much, if a read sequence is of 100 length, so if it is aligning to the reference genome and 100 matches are there, it means the complete match is there of the read with the reference genome, right? Then this is not important for you because this is doing the same thing, as because I have used only the single read file, not the paired and read files. If there is the paired and read file in these three columns, you will get the result. It means if there is zero or hash steric, it means that there is no mapping of the paired end, right? So zero and asterisk means there is no mapping of the paired end. Then there is a sequence of the this is the sequence actually of the read, and this is the quality score of the read, right? These are actually the main columns are these only till the cigar. So this is the main 4 5 columns which are important for you to understand the read is mapping to the reference or not. Now you here you can see also one more thing. Cigar, that 100 m is 100 matches. If there is if a unit is like written as 50 m 2d 50 m, so it means 50 matches, two bases were deleted, and then 50 more base matches. Okay, 50 m, two deletions and 50 matches. Now since you have understood this, next is BAM. This BAM is uh, completely similar to the SAM. The objective of the BAM and SAM is same. They have to show a sequence where the read ID is aligning to the reference genome. So here you can see the only difference between BAM and SAM is that BAM is non-human readable text file. Only computer can read it because it's a binary file. So here it's written binary file. Normally it's happen in computer that any file is converted when once it is converted into binary version, then this that file become very small in size and also the speed increases. Therefore the BAM is much better and more efficient than SAM, but you cannot read the BAM file. This is the only difference between BAM and SAM. Then after understanding BAM and SAM properly, so again see the format properly, I'm going to ask the question. Now how you have to think for this format as an bioinformatician. So you can click it also. Now now think like a bioinformatician, and there is a first question here. Look, what does this cigar string means? Oh, that I have just told you. So answer it. If you see something like this in the file, how you are going to interpret the answer?
Ma'am, 50 bases match and two bases inserted. Then 48 more bases matched.
Very, very good, Swiksha. Okay, correct. Actually, you are correct. So this is the way you are going to understand how many bases have been matched to the reference genome. So 50 matches, 2 insertions, and 48 match. Now the next question is, if you see a line where the third column is a static and the fourth column is a zero, in this read mapped, or is this read mapped or unmapped? If you have
Ma'am,
taken the
there is no mapping because there's no mapping because of the like paired end नहीं हुआ है, सिर्फ एक single read लिया है तो
no
asterisk और zero है.
okay, so what you are saying, it's regarding the paired end, but I am asking the in the third and fourth column. If you have in the third and fourth column, so here is the third and fourth column. This is the first column, second and third and fourth. And so they are saying in the third column, if you find an asterisk, and in the fourth column you find zero, then also it means it is unmapped. You guys are correctly saying, Ishita Pal, Anumita, Ariba, all of you are answering it very correctly. That in the third, unmapped is the answer. But in the third and fourth column, if there is a static here, because if a particular read has not mapped, it won't show the ID of the reference and won't show any position. That's why it is the answer is unmapped. Nice. So the next question, since I'm not going to give you a lot of questions here, so I have just told you the format. So you are going to use a tool, SAM tools, to work with the SAM files. So you will understand in the hands-on how SAM tools will help you out to understand each and everything in the that tool makes easy to understand the mappings. Last format is the VCF format. VCF format is the full form is variant call format. This format is a standard output of a variant calling pipeline. It specifically records the positions where a sample differs from a reference genome, identifying SNPs, insertions, deletions, and structural variants. This means when you have the reference genome and you have your read files and you have mapped it also on the reference genome. Now the positions where there is no the mapping has not occurred or there is some unmapping happened, so those are the points where mutations has actually occurred. It's not always that mutations has already occurred. You also have to keep some thresholds to confirm that at this position you are facing some mutation. So mutation can be SNPs, insertion, deletion, structural variants. In COVID also, if you have seen, multiple variants have been formed. In that case also, what people used to do, they take the sample of the patients, sequence it. After doing sequencing, they get the reads and they align those reads to the reference COVID genome, and they find the mutations at the point and find the mutation between the reference and the reads, and they come to know at what points actually mutation is occurring in different regions. So VCF file is actually giving you the information related to data analysis. You can analyze this information to get significant results and for the publications. So VCF file, structure of VCF file is the file is like this. It has two hash in the starting. These are the if there are two hashes, it means these are all are giving the header information where they are going giving you the version, what is the version of the file format, what is the reference ID used, what is the length. So all this information you will get when there is at the start where there are two hashes, right? But in the but in this column, in this row, you can see in the fourth row there is only one hash. If there is only one hash, it means this is the main header line. So in the header line you can see there is chromosomes. The name of the chromosomes are listed. Reference chromosome, this is the reference chromosome, this is the position, this is the ID, reference, this is the reference nucleotide present in the reference genome, and this is the altered nucleotide present in your read dataset, in your contigs. Quality of the score, and this is the filter passed, and the info depth is also telling you to what extent this particular mutation is has a high confidence or not. So for the high confidence you can see DP 30 plus will give you high confidence, and DP equals to 2 will tell you that it's by random chance or some error. So in this way you can understand where actually the at which position of the reference the mutation is occurring between reference and alt. So by seeing this, can you answer me this question? Don't unmute. Please don't unmute yourself. Just write 1, 2, 3 for each position. Write in the chat box.
Hello, ma'am.
Don't unmute yourself.
Ma'am,
Write in the chat box so that everyone can understand what can be the answer for this. For each, if you have any query, you can ask. But
ma'am,
for to
sorry,
give the answer, you have to give in the chat box
ma'am, sorry for interruption. Can you repeat that? Uh,
the question.
wala?
Okay.
No, no, ma'am. VCFL actually I was disconnected.
This one?
Yes, ma'am. From the starting VCF.
Okay, from the starting. Okay, I'll
Yes.
explain it again. Um, so this is a VCF file format, right? In this complete format, there are two hashes in the three columns. These three columns are only giving you just the information that in these three wait for the answer. Okay, so in these three columns there are two hashes. So you can see NC is the reference genome. They are giving the information in which reference genome the reads have been aligned. So this is the reference genome, this is the length of the reference genome. This information is giving you that the work has been done past, and this is giving you the version of the VCF file. So these are not that important. The important one is the where there is only one hash, which is in the header. You can see there is only one hash, right? So here you can see in each column what information you are getting. In the first column there is the reference genome on which you have used your data to analyze the point mutations, to analyze the mutations. So this is a reference ID. This is the position where actually the changes have occurred from the reference genome. This is the reference nucleotide. So in the reference genome at this position there is a nucleotide A which has altered to G in your current data. Then you will see the quality. So A to G alteration has occurred and the quality is a good quality score 60. And you can also see the depth is 25. This is almost fine, not that very good, because 30 plus is taken as to be very good quality score. And so here you can see there is an alteration from reference to alt. Now on the basis of this you can see the mutation here. Can you, on the basis of this, uh, can you answer me this question? So now you can answer in the chat box. If you still have some query, you can ask. Bingo, bingo, bingo. You guys are correct, are answering very correctly. Everyone is answering so correctly. I hope you have understood this. If not, I will explain for whom who have not understood. But I think almost everyone has answered very correctly. One Gargi has written 1011 greater than one base replaced by another, so SNP. Oh, okay. So now you can see here, here in from C to reference in the first column, first row. Sorry, in the first row, you can see at this position 1011, at the from the reference nucleotide C has been changed to T. So it is a point mutation, SNP, or some people have written it as substitution. Next is 1045. This is the position where A has changed to ATTTC. It means a lot of nucleotides have been added. That's why it is called insertion. And in the third one, at this position 1089, GTC has been reduced to G, only G is left. PC has been deleted. So it is a deletion. So everyone has answered very correctly. I am happy to see this. Ma'am, can you explain please explain what is DP? So DP is the depth at which actually what happens when a sequencing machine search for a particular changes in the in your read sequences. DP is telling to what extent this value is of a good confidence. Is it of good confidence or is it of less confidence? So should you consider it or not? If DP is good, if DP is more than 30 plus, it means it's of good quality. If it is around less than 30 or very less than 10, so it is of low quality. So it means you have at each and every step you have to figure out that results which you are getting are because of some error or because you seriously getting some findings. Quality and DP. What define qual is mapping quality. So here you can see the quality means C is converting to T. This is what I actually depth is quality is C is moving to T, and this is a quality score instrument gives that yes, till 60 is the maximum quality. So C is converting to T, it's a good quality. But to what extent you can confirm this that it's a good quality, you can see the DP score. Got it? Shivangi, do you got the answer? Okay. So my next question is this. Finally, you have completed all the 5 formats. Just answer me in the chat box. What is X, Y, Z, and M? Answer in one go. Give all the answer in one go. You guys are not writing spam. Sam is fine. So many of you are not answering correctly. Some Arijit has answered actually correctly, and someone else also. Fast Q, Fasta. Minu also has answered. Minu has the correct. Minu has given the almost correct answer, completely correct answer. ZM. So I have seen everyone's answer now. I'll let you know why someone can tell me why you are thinking that X is a FASTA sequence. Why you are thinking X is a FASTA sequence. After from the sequencing, you get an output. From the sequencer, you get an output, raw reads. And that can be faster. Earlier in the earlier sequencing, like sequencing, if I say in the Sanger sequencing, in the initial at the initial level, FASTA was a format, output format, but now onwards, FASTA is no longer a format for any sequencing machine. So raw reads are actually the FASTQ files now. From the sequencing machine, you will get X raw reads as FASTQ. Then in the Y is the reference genome. Reference genome is completely aligned, and there is no need for the reference genome to be. Reference genome is made reference genome when it is completely annotated and has been experimentally proved. So there is no need of quality score in that. So always a reference genome, you find a reference genome a complete, because read is of optimum length. It's of 100 base pair to 150 base pair or around 300 base pair, but reference genome cannot be of a particular 100 to 300 range. It has to be completely around in MBs, in the length can be in MB of the reference genome. So reference genome, why can all cannot be a FASTQ, it has to be a FASTA. FASTQ cannot store so much of data like in a one stretch, but reference genome has to be stored in FASTA. Next is aligned reads Z. So Z is here SAM or BAM both will work. It's not like that only SAM work or BAM work. Both files are same, so SAM and BAM both will work. Next is variant discovery, where you are going to see that the variations are occurring, and this is VCF. Almost VCF, though everyone has given rightly. The problem is with the starting ones only. So I hope you have understood by what is X, Y, Z, and M. And if anyone has any question, so can ask me. Do anyone has any query, or is everything? Yes, yes,
Ma'am,
yes.
here can X be faster file also?
X be a faster file.
Yes, I'm asking, can it be a faster file or can it only be fast queue?
Okay, for that I answered that initially when there is only Sanger sequencing, at that time there the output format of the raw reads was a FASTA, but now onwards FASTA has not been used because it does not include quality score. So the current sequencing procedures the current sequencing procedures which are used are Illumina sequencing, nanopore sequencing. So these are giving the FASTQ files only. So X cannot be it's partially, but it's FASTQ only.
Thank you, ma'am. I got disconnected in between. That's why I have.
Okay, no issue. So anyone has any query now? Or okay, fine. So all these five formats, I was just giving all the five formats. So I hope you have understood everything. And if you have understood, so I'm just I just hand over this Zoom information to Zoom screen to Ajay. He's going, Ajay sir, yesterday he has told you about everything grep and all, but he was not able to take the quiz because of the time lag. Now he's going to take the quiz, short quiz of 15 minutes. So be ready for that. I hope you have understood everything. Is